In South Carolina, Susan, Bethany, and Asia experienced their first flood and decided to go for a swim and drank the water from the faucet when there was a water advisory. Now they all have a bacteria infection called cholera. How will their immune systems fight the parasitic worms? All immune systems have proteins called complements that will undergo a classical pathway to form the MAC complex. In the case of Susan, Bethany, and Asia contracting parasitic worms from contaminated water they swam and drank during the flood, the classical pathway will activate when the antibody binds to the antigen, which in this case is the parasitic worms. The purpose of the classical pathway is to form MAC, which stands for membrane attack complex, that lyses the membrane of a target cell. In other words, the MAC complex will hole punch the membrane of the target cell. The initiation of the classical pathway will begin with the cascade of complements that will form the MAC complex, resulting in the parasitic worms being hole punched, which will eliminate the bacterial infection they have all contracted. Here is a figure that shows all of the complement proteins that are involved in the classical pathway. The players involved in the classical pathway are the antibodies IgM that will bind to the antigen, or IgG that will bind to the antigen. IgM is the best antibody to use because when it is secreted, it is very big. Because of how big IgM is, it undergoes conformational changes when it binds to the antigen. It goes from a planar formation to the staple formation. On an aside, because IgG is a monomer, it makes it less efficient for the binding of the C1. C1 must bind to a cross-link of IgG, which just means it needs to bind to at least 2 IgGs that are close together. Another player for the classical pathway is complement 1, or C1. It is consisted of 3 parts: 6 stalks, which are the Qs, 2 Rs, and 2 Ss. All together forms the biggest complement, C1. Some more players in the classical pathway are C2, C3, and C4. All of these complement proteins will be cleaved into big and small portions. For C3 and C4, the big portion is B and the little portion is A. For C2, the big portion is A and the little portion is B. C5 is another complement protein in the classical pathway that will be cleaved into a small and a large portion. C6 and C7 are 2 other complement proteins. The large portion of C5, also known as C5b, will bind with C6 and C7 to form C5b67. Other proteins involved in the classical pathway are C8 and C9. C3 convertase is just C4b and C2a binded together. C5 convertase is just C4b, C2a, and C3b all binded together. Then C5b678 is the addition of C8 from C5b67. Another very important player in the classical pathway is the antigen or the microbe, which in this case is the parasitic worms for Bethany, Susan, and Asia. The classical pathway is antigen dependent, which means it depends on the presence of an antigen in order for the pathway to be activated. Lastly, C9 is usually prevalent in an abundant number of C9s, usually about 10 to 16. C9s are what form the pore of the MAC complex. The poly C9 pore binds to the C5b678, which then binds to the membrane. That full complex is what will lyse or hole punch the parasitic worms. The activation of the classical pathway begins when the antibody binds to the antigen and then C1 binds to the antibody. Once the classical pathway has been activated, C4 moves towards C1 to be cleaved into C4a, which leaves to do other great things in the immune system, and C4b binds to the microbe. Once C4 has been cleaved, C2 goes to C1 to be cleaved as well, to form the small portion, which is C2b, which leaves to do other great things in the immune system, and C2a moves to the microbe and binds to C4b. C4b and C2a form C3 convertase. C3 convertase cleaves C3. This is an amplification step, which means it can cleave several numbers of C3 into C3a, the small portion, which again leaves to do other great things in the immune system, and C3b binds to C3 convertase to form C5 convertase. C5 convertase cleaves C5 into C5a, which leaves to do other great things in the immune system, and C5b. C5b binds with C6 and C7 to form C5b67. C5b67 binds with C8 to form C5b678. Then an abundant number of C9s come to bind with C5b678 to form a pore, which then binds to the membrane. We'll then move to the parasitic worm and lyse it or hole punch it to kill it and get rid of the bacterial infection. The end.